Hello, if you're new to these videos, my name is Erica Bowen. I'm a graduate student at the University of Georgia working in the Mammalian Lab, and I am making a series of videos on how to do cell culture techniques with adherent cells for training my mentees and training students that I have in a class that I'm teaching this semester. I've already made a video on how to aliquot, how to passage cells, how to thaw cells, and how to make media for the cells. Today, I want to talk about how you would do a cell count. In this video, I'm going to be showing both kinds, manual and automatic. Manual cell counting is starting to become a little bit obsolete. However, during my academic career, I've had to manually count and make the switch to automatic counting. And I would say our lab is like pretty up to date on stuff like that. So it's very likely that you might need to know how to manually count still, and maybe for a little while longer. But also, I think it is important to learn how to manually count because that process teaches you the math and what's going on behind the automatic counting, which is extremely important, especially when determining the dilution factor. And I'll talk more about what that means. So yes, you can estimate the amount of cells that you might have in a flask or for an experiment, but it's not best practice. You should know exactly what's going into your experiment at the starting point to ensure that your data points and your replicates are all starting from the same spot in the growth profile. Estimation is prone to enough error to make some significant impacts on your data after the fact. So very important that you, when you're doing real experiments and not just growing out a cell line, you count the cells at some point at the beginning or at the end. And cell counting is done once you have some sort of cell suspension. So I think in the description below, I'm going to link in the passaging video the point at which you would want to sample for a cell count. It is highly likely that you are going to grow out some cells and then you're going to passage into the vessels that are going to be used for your experiments. Now, I prefer to do a cell count with the cell suspension before it enters the centrifuge during passaging, so that while the cells are spinning down, I can do a cell count, and then hopefully once I'm done doing the cell count, my cells are done spinning down, ready to be resuspended in fresh media and split into the flasks or well plates that they're planning on going in. Something else I also like to do is if it's going to be some sort of complicated math involved with that cell count, like once you get the cell count, you need to like split it in some crazy ways. I make Excel sheets to do that calculation for me before I come into the lab. So for example, I have an Excel sheet where I take cell count, I calculate how many cryovials I could possibly get from that cell count, and then it auto-calculates how much freeze media I should make to resuspend the cell pellet in and then split into the cryovials. I have other Excel sheets too where I take the cell count, I input it in with formulas, I calculate how much I want to seed a flask with. So the user inputs seeding density, what the volume was that the sample was taken from, and then takes in a cell count. And then the user can also input like desired cell density in the flasks or the well plates that they're working with, and it'll auto-calculate all of this stuff for me. I highly recommend this way of doing things, especially if you're someone that isn't very great at math, especially in the beginning, like when all of these things take longer for you to do. You may start to feel an urgency and feel rushed trying to do the math. If your cells are sitting in the hood waiting for you to do something with them, it can be stressful. So I highly recommend doing an Excel sheet, or if you don't know how to use Excel, honestly, like just half work it out on a piece of paper in a way that you can just like plug in a number later. Just make sure you know what math you're about to do with that cell count before you have cells like waiting for too long to be used. For both cell counting techniques, automatic and manual, you're going to start with the same steps. So let's say you did all the things in the passaging video up to the point of the time that I've linked in the description below. And then you have your cell suspension. You're gonna wanna take a 100 microliter sample from that cell suspension. This is small enough so that you're not disrupting like the total volume of your cell suspension. You don't want to sample like half of it out, that's way too much and you've lost a bulk of your cells. So just 100 microliters, and you put it into a sterile microtube here. I've taken out one and put it in a tube rack. And autoclaved microtubes are kept in the biological safety cabinet. So in here is my 100 microliter sample. A bunch of those are kept in our biological safety cabinet in the mammalian lab. So what I would want to do is, simultaneously as I'm going to bring my cell suspension to the centrifuge to spin it down, I'm also going to be counting this at the cell counting station. In the mammalian lab, our cell counting station is located next to the microscope. I bring the whole tube rack out with my sample and place it here. I need yellow pipette tips. I will also need Trypan Blue, which is just kept here for use. This Trypan Blue is not sterile. It's kept and used outside of the biological safety cabinet. But you still want to be like wearing PPE while you're doing this whole thing, because the cells are biosafety level 2 hazard. This Trypan Blue is a carcinogen and a mutagen, so you don't want to get this on your skin. You'll also need a micropipette, which is located on the opposite side of this bench. You need a micropipette that could measure out 100 microliters and one that can measure out 10 microliters. This one can do both. So I set it to 100 microliters here, and I'm going to add 100 microliters of Trypan Blue to the cells. So I always try this first, 100 sample and 100 Trypan Blue. This gives you a dilution factor of 2. Then you're going to change this micropipette to say 10, because this is actually how much goes into your cell counting chamber on the cell counting slides if you're doing automatic, or in the hemocytometer for manual counting. I'm going to manually count first so we can talk about the math before going into automatic counting. A hemocytometer, 2 clean ones are kept by the sink in the mammalian lab, and that is the hemocytometer itself, and a glass slide as well. Be very careful not to break these. So I bring that over here. I'm going to go ahead and close the Trypan Blue because I don't need it anymore. And I'm also getting a little paper towel to put this whole hemocytometer on, just in case I spill Trypan Blue or my little sample. The paper towel can catch it, and then I can throw it away in biohazardous trash. With the hemocytometer, you want to make sure this glass slide is clean. This one's pretty clean, but if it wasn't, you can get a chem wipe and further wipe it off. This lays and aligns itself perfectly with these hemocytometers. So the long side aligns with the long slide. And you want to make it so like the center of the glass slide is laying across the center of the hemocytometer here. So this is pretty straight. And now we're going to take a sample of the sample. So I have my little microtube here. I'm measuring out 10 microliters, kind of pipetting up and down to get a good sample. And I'm balancing this on my pinky and injecting this into the entrance of this channel. When I'm doing this, I'm not touching the hemocytometer at all, but the tip is inside the channel. So let me see if I can actually show this. Like I'm placing the tip in there, but I'm not actually like moving the slide or anything like that, or touching down. So that's why you might need to use a pinky if you're like extra shaky like me. For experiment purposes, I always run both sides of the hemocytometer. So technically you could do a cell count off one side, but you should, for good practice, be counting both sides and then averaging both sides to get a more accurate cell count. Now, I've been doing this for a while, so I was able to pipette like without any bubbles appearing in the squares. If any bubbles appeared, you would have to redo that, which is why we have the second hemocytometer. You could just go over there and retry on that other one until you get two sides that aren't compromised. So now I'm ready to count. The cell counter is located in this drawer. This little fellow right here. And I set it to say 0 to start. And now I'm going to bring the hemocytometer to the microscope. Place it so it's as straight as possible. And I'm going to look for, in 4x, the center of the hemocytometer, which I'll show on the screen what it's supposed to look like. And the area where you see like a bunch of tiny squares here. And there might be some fibers from your like Kim wipe and stuff, and sometimes you get these like black spots, which is crystallization that's happening. That means I need to filter it. But these tiny squares here, where like all the grid lines are converging, is the center of your hemocytometer, which means this up here is your top left square. And then all the way over here is your top right square. And each of these squares have a 4x4. They're made of a 4x4 of squares. I zoomed in a little bit closer to show you. But how Trypan Blue works is that it dyes the background blue, and any living cells are going to be this yellow color here. Any dead cells will not be as bright as the living cells. And you would only count the really bright yellow cells like those. So once I find that top left part of the hemocytometer, I focus my microscope, and I am only counting cells that are glowing. So actually I've never done this before, but I'm actually doing a count on cells that I just thawed. So I don't actually think these are dead. Like maybe they're just not reacting with the Trypan Blue as expected because they're freshly thawed. But some of these cells are. So like if this was a typical cell count, I wouldn't count that one as alive, but it might be. Whereas that one that's glowing pretty brightly, that one is definitely live. That would be a count. This is also a good opportunity to show you cells that are on like the edge of a square, like that one, I would count as being inside the squares as well. So I'm just gonna go through and count these for you to give you an example. So this is the top of the top left corner. 1, 2, 3 on the edge. 4. So this is telling me there's 4 cells there, which technically, to be an accurate cell count, you would want to see 15 to 50 cells per corner of the hemocytometer. So if this was like cells that I just passaged, there would not be enough cells for me to get an accurate count, which would not be good for the experiment, and I would continue growing them out until I knew that I could get an accurate cell count. As you count one square, you should be clicking this. So like 1, 2, 3, 4. And then when you want to like go to a new square, spin this back down to 0 and start counting again. When there's a higher amount of cells there, like if there's like a near 50, this is really helpful to like speed count and not lose count of your cells.
Hi, this is Erica from home here to interject. So real quick, if you were counting your cells and you got above 50, you would want to change your dilution factor. So if you counted a square and you got 100 cells, you could up the dilution factor to 4 and recount the cells using Trypan Blue. So if you initially added 100 microliters of Trypan Blue, you could add 200 microliters more to have a total volume of 400 microliters. And the dilution factor would then be 400 divided by 100 microliters, giving you 4. This dilutes your sample to bring it into the appropriate range, but then you would also need to account for that in the cell counting formula that calls for a dilution factor.
So I'll probably do like an example numbers from home because I don't really care about the cell count and it's late at night. But once you're done counting the 4 squares of one side, you would want to move over to the other side of the hemocytometer. Again, find where those squares are and go ahead and count through that as well and get an average.
Okay, now to actually address this cell counting formula that I've been talking about through the video. So once you have cell counts for all 4 squares, each side of the hemocytometer, average each square altogether to get an average count per square. You should already have a dilution factor determined from the formulas I've been showing throughout this video. And you will multiply this by 10 to the 4th. This 10 to the 4th is related to the area of the square, which is 1 millimeter by 1 millimeter. It works out so each square has a total volume of 0.1 millimeters cubed or 10 to the negative 4th centimeters cubed. And this is where this 10 to the 4th is coming from. Once you have a count, you should be able to calculate cells per milliliter. So all of this is giving you a cell concentration. This is still not the total amount of cells that you have in your cell suspension. So to get that, you must multiply it by the original volume of the cell suspension. So whatever your sample came from, that volume multiplied to the concentration. That'll give you the total number of cells in the cell suspension. So let's work through a problem together. You have a cell suspension of 25 milliliters that needs to be counted. A 100 microliter sample of the cell suspension is taken and mixed with 100 microliters of Trypan Blue. 10 microliters of the mixed sample is counted using a hemocytometer. The following number of cells were counted in the 8 squares of the hemocytometer, so both sides. 33, 32, 36, 29, 31, 31, 30, 40. What is the concentration of the cell suspension and what is the total number of cells in the suspension? So you have 2 questions here. This is the answer here. So to get this answer, you first average the count. Right off the bat, I'm seeing that these numbers are all very similar to one another. So another cell count doesn't need to happen. If they were all very different, you would want to retake that 10 microliters, put it in a clean hemocytometer. The average of those 8 squares is 65.5. You need to find the dilution factor, which in this case is found by adding Trypan Blue volume to the sample of the cell suspension volume. You'll get 200 microliters. Divide that by the 100 microliters of the sample. This gives you a DF of 2. Next, you can find the cell concentration. So using the formula I showed before, 65.5 times 2 times 10 to the 4th, this is how many cells per milliliter you have in your cell suspension. Then to find the total cells in the suspension, take that number and you multiply it to what the sample came from, which was 25 milliliters total. This gives you how many cells are in the cell suspension, which is 32,750,000 cells.
Now I'm going to show you what automatic cell counting looks like. So pretend like we didn't count that at all. We keep cell counting slides out next to this automatic cell counter that's located here. You turn it on the back. And unlike that hemocytometer, these are disposable. So once I'm done counting on this, I would put it in the biohazard. For this one, same thing. 10 microliters we're going to measure out. And it looks pretty much just like the hemocytometer that we manually count with. There's an A and a B side, and where the arrow is pointing is where you eject the 10 microliters. Unlike the manual counter, it seems like it's okay to actually like touch the tip down inside this channel. So it's a lot easier to actually pipette your sample into this. And I'll show you. So I'm just gonna pipette into one side and just do a count on that one side. This packaging can go in normal trash. And with the side that you'd like to count, put that into the front of the automatic cell counter, and it plugs in kind of like a floppy disk, which I know is a really dated way of explaining that. I can't think of anything else to like describe that as. But there's a click when it actually goes all the way in. And the first thing it does is establishes a bright field image where it autofocuses on the cells. So you can actually see this focus pretty good on the cells. You hit count, and you would go by the live cell count. So again, I just thawed these cells, so I think they're not reacting with the Trypan Blue well. There's also crystals that I need to filter out of this bottle here. But let's pretend for a second this was your actual cell count. You would go by this green bar here, which tells you how many living cells are in this cell count, and it's giving you a concentration. So it says that there are 7.32 times 10 to the fourth cells per milliliter in this sample. In the red, it's telling you how many it's counting as dead. It's also showing you what it's circling as dead. So it's obviously circling some of the cells that aren't lighting up as much, and it's circling like some of the crystals too. But I'm not worried about this. Up here, it gives you like the uncorrected total of both of those. If you wanted, you could go by this total as well for your experiments, especially if you just had a lot of live cells and you suspected that a lot of the stuff it was counting as dead is debris. And you can usually scroll around in here and see and make an educated guess. You can save this data to a USB if you wanted to. There's a USB port also located at the front. This could export your results as TIFF files, the images as TIFF files. You could do a PDF report, Excel, whatever. I've never done that. I've always just taken that number off the screen and done my calculations with that. And then once you're done with cell counting, you can eject this out by pressing on it and placing it in the biohazardous trash nearby. Same thing with your sample. If you no longer need it, it also goes in the biohazardous waste here. So now I'm going to clean up. And clean up is turning this off, putting everything back where you found it. All of this stuff goes back. Trypan Blue is closed and placed back up there. This goes back on the other side of the bench. Very important, this hemocytometer needs to be cleaned correctly, which means you take the glass slide off, you bleach it, ethanol it, and then put DI water on it. Sometimes I just do ethanol. I think that's sufficient. But as long as you are doing something to kill or deactivate the biologics that are on that. So ethanol bottles kept at the sink here. I'm spraying off the glass slide with ethanol. Then with DI water also kept in a wash bottle by the sink. You can use a chem wipe to dry it off. And then place it back where you found it, which is there. Same thing for this. Ethanol, make sure to get in the channels. DI water, dry this off with a chem wipe, and place it upside down onto this folded chem wipe here. You should never be cleaning the hemocytometer with sink water because there's too many hard metals in the sink. I recovered the EVOS. I'm going to put the cell counter back in the drawer. And that's good for cleaning up the cell counting station. If you get really good at that, you should be done right as your cells are done spinning down. And then at that point, you have a cell count, you know what you want to do next, hopefully through your Excel sheet, and then you can continue working in the biological safety cabinet.
Okay, now I would like to walk through a little bit more of a complicated and more applicable cell counting and flask seeding example. So you need to expand cells in culture for an upcoming experiment. You remove a 100 microliter sample from your cell suspension and mix it with 100 microliters of Trypan Blue for counting. After counting your 5 milliliter cell suspension, you determine an average of 55.5 cells. You need to seed 3 T75 flasks with 1 million cells each. What is the total number of cells needed for the 3 flasks, and do you have enough cells to carry out your experiment? You will need 3 million cells for the flasks, and yes, there is enough here, and I'll walk through how to do that. So first, you can find the dilution factor, which is the same as the previous example and same as the video. 100 microliters of Trypan Blue plus 100 microliters of the sample cell suspension divided by 100 microliters of the sample cell suspension will give you 2. You can next find the cell concentration. It gives you the average count already, which is nice. So you can just plug it in. 55.5 times dilution factor, which is 2 times 10 to the 4th. It gives you 1,110,000 cells per milliliter as your cell concentration. You can then find the total cells in the suspension by multiplying this value to the volume of the cell suspension that you sampled from, which is 5 milliliters. This gives you 5,550,000 cells. This value is greater than what would be needed to seed 3 T75 flasks with 1 million cells, which is just 3 times 1 million, which is 3 million. This would make it so that you have 2,550,000 cells remaining. So you would have enough to carry out this experiment. So just to continue working it out, based on what we determined before, how much cell suspension do you need to add to each flask if we're seeding those 3 T75 flasks with 1 million cells each? We would need 0.901 milliliters of cell suspension. And then this is where we employ the C1V1 formula. So we know C1 from the previous problem. This is our cell concentration. This is what we counted. So 1,110,000 cells per milliliter. V1 is what we're solving for, how much cell suspension to take from that cell suspension we counted. And then C2 is a little more tricky, but this is the destination's concentration. So we know the working volume of a T75 flask is 10 milliliters, and we want to put 1 million cells in that flask. So you can simply divide those two values to get a concentration expressed, where this would be 100,000 cells per milliliter that would need to go into the T75 flask. And then this is undone pretty much by multiplying by our working volume of V2, 10 milliliters. If we were already pre-soaking these flasks, like with 5 milliliters, they already had 5 milliliters in them, then you would plug in 5 milliliters here as the additional 5 milliliters that would need to be added to the flasks that already have 5 milliliters, if that makes sense. So like if the flasks were pre-soaking in 7 milliliters each, you would put 3 here, because 10 minus 7 is 3. But anyways, continuing on, solving this would give you that V1. And then furthermore, if you're adding this cell suspension to a T75 flask, you would need to add 10 milliliters minus 0.901 milliliters of fresh media to bring that flask to the working volume. Now, thinking about how you would actually logistically do this, because maybe you haven't thought that far. So this cell suspension that we calculated here is based on the fact that we sampled from 5 milliliters of media. If you want to use that number, you need to resuspend that cell pellet in 5 milliliters. But what happens if you can't do that? So one scenario could be that you ran out of media. You only have 2.5 milliliters of media left. You can't resuspend in 5 milliliters. Well, you can resuspend in the 2.5 milliliters, and that's half the amount that you intended to resuspend with, this would make it so that you need half the amount of cell suspension, which is 451 microliters, or 0.4505 milliliters of cell suspension. A situation when you might also want to do that is if you just want to be more conservative with the amount of media that you're using in your lab for whatever reason. Another example could be that you had a really low cell count. So let's say you got straight 10s on your hemocytometer. This would give you a cell concentration of 200,000 cells per milliliter. Now let's say you wanted to actually see with 300,000 cells per milliliter in a T75 flask that already has 5 milliliters of media in it. Well, this is saying that you would need a V1 of 7.5 milliliters. But 7.5 plus 5 is greater than 10 milliliters, which is the working volume in a T75 flask. To bring that value down, you can instead resuspend in 3 milliliters, which is 5 times smaller than intended. And this would make it so that your V1 is now 1.5 milliliters instead. So this added to the 5 milliliters that's already in your flask isn't going to exceed the 10 milliliter working volume. And then you can add the additional fresh media desired, which in this case would be 3.5 milliliters. So I hope that all makes sense, and I hope that this video helps you in cell counting and doing calculations pertaining to that. Thank you.